Today we're delighted to have two speakers with us that are going to be talking about a common subject. And Andrew, you can go ahead and share your screen if you want. There of cisco, Coregonus artedi, and the Great Lakes. And we know historically they used to use a diversity of habitats. And we think in many of the lakes, not only in some of them, sadly, they're extirpated, but in many of the Great Lakes, we know they used to use all of these alternative habitats. And now we're beginning to find evidence, and I think you're going to hear more of that today about them using some of these unusual or returning back to some of these habitats. So tributaries and drowned river mouths. We're really excited to hear from Andrew Honsey and Ralph Tingley, both from the USGS Great Lakes Science Center. So Andrew got his PhD from the University of Minnesota, working with Paul Venturelli, got his master's degree working with Thomas Hook at Purdue University, and his bachelor's degree at Hillsdale College here in Michigan. He did a postdoctoral appointment with Gretchen Hansen, also at the University of Minnesota. And then we were fortunate enough to hire him in 2020 as a fisheries biologist. He is focusing on Coregonine restoration and based at the Hammond Bay Biological Station up in northern lower Michigan. And so Andrew will go first today, but I also want to introduce Ralph because they're essentially going to do a trade-off, a passing of the baton. Ralph, without me coming back on to interrupt them. So Ralph got his PhD at Michigan State University and also his master's at Michigan State University. He came to Michigan, however, from the University of Connecticut. So coming from the East Coast where he worked at, got his bachelor's in natural resource management, focusing on fisheries. He did a postdoctoral position at the University of Missouri, USGS Missouri Co-op with Craig Pockert. That was 2017 to 2020. And just as with Andrew, we were really pleased to recruit Ralph to the Great Lakes Science Center in 2020. And he is based at our Ann Arbor Great Lakes Science Center office. So guys, thanks again for offering to do this. They've gone through this already today, which is really impressive, doing a practice talk for everybody. It's about 50 minutes, and hopefully our great audience will stick around for questions and discussions as we always do. We'll probably have you ask questions of Ralph first because he does have another appointment that just came up that he'll have to make. But I know that Andrew can stay on closer to 2 o'clock if the discussion goes on that long. So thanks everybody, and we look forward to your talk.
Excellent. Thank you very much, Bo. And yeah, thanks to everyone for taking the time to listen to Ralph and I today. Really quickly, can you all hear me okay?
Sounds great.
Great, thanks. So as Bo mentioned, we'll be talking today about seasonal migrations of cisco in the Great Lakes to both tributary and drowned river mouth habitats. And I'm going to begin by briefly touching on the history of coregonines in the Great Lakes. As you may already know, coregonines have declined across the Great Lakes over the past couple of centuries. For example, if we just look at the ciscos or subgenus Leucichthys within Coregonus, we see that the Great Lakes historically maintained a wide diversity of species, many of which were among the most abundant fishes in the lakes. However, over time, these fishes declined due to overexploitation, habitat degradation, and interactions with invasive species, among other factors. And many species are now extirpated from some lakes or thought to be extinct. So today, what we're left with is a mere fraction of the coregonine diversity and biomass that we once had. So in an effort to promote resilient fish communities and sustainable fisheries, Great Lakes managers are now interested in restoring coregonine diversity and have endorsed this adaptive management process for doing so, which we call the coregonine restoration framework. For the sake of time, and since you may have heard about this framework in previous presentations in this series, I'm not going to spend much time on it today, but suffice to say that it provides a structure for planning, implementing, and evaluating conservation and restoration actions, including things like fish stocking and habitat restoration. One key element to the success of these conservation and restoration efforts is a good understanding of habitat use. And this understanding can come into play in many ways. For example, if we're conserving or restoring habitats, we need to know which habitats the fish use so we can target the right ones. We also need to understand habitat use for fish stocking decisions so that we can know where and when to stock fish to maximize the probability of success. And spawning habitats are often the focus of these types of efforts, given their importance to fish populations, but other habitats like key foraging areas or migratory corridors can also be important. And indeed, this element of understanding habitat use shows up explicitly in the Cregonian restoration framework, as one of the science planning steps is devoted to describing and mapping populations and the habitats they use. For Cregonians in the Great Lakes, understanding habitat use is somewhat complicated by the fact that many of these fishes migrate. For example, many populations of cisco, Coregonus artedi, migrate from offshore waters to nearshore areas to spawn. And historically, these migrations provided opportunities for fishers to catch large numbers of cisco in really shallow water, including along beaches using seines as shown here. And contemporary fisheries for cisco also target them in and around the spawning period. In addition to migrations within lakes, Cregonians also migrate to other habitats such as rivers. For example, lake whitefish historically migrated to rivers in many areas to spawn. And though a lot of those river spawning runs have been lost due to habitat degradation, we're now seeing whitefish recolonizing rivers in some areas, particularly in tributaries to Green Bay in Lake Michigan, which is an exciting finding that shows that these fish can utilize previously degraded habitats. I'll note quickly that although both of these examples highlight spawning migrations, Cregonians may also migrate for other reasons, like habitat suitability or foraging. These migrations have some important implications for conservation and restoration. For example, following Schaefer and Stein's conservation pillars, which are often called the 3 Rs, migratory Cregonians may represent key aspects of diversity, including genetic, phenotypic, behavioral, or functional diversity that are worth conserving or restoring. Migrating populations may also contribute to species-wide resiliency to stochastic disturbance events, and conserving them or using them for restoration, such as stocking in other locations, can maintain or increase the species' ability to withstand catastrophic events through redundancy. Migratory populations can also contribute to the stability and resiliency of metapopulations and communities through portfolio effects. So for example, populations that spawn in multiple distinct habitats like rivers can have differential recruitment success across years that can act to stabilize metapopulation dynamics. And of course, the classic example of this is the Bristol Bay sockeye salmon fishery. In addition, some migratory populations may have more consistent recruitment success than others, and this can lead to broader age diversity in the metapopulation, which is another way that these migratory populations can provide a portfolio effect that acts to stabilize dynamics. And finally, migratory populations can act to couple habitats and influence ecosystem nutrient dynamics. One great example of this can be found in the work by Stockwell et al., who showed that spawning migrations by cisco in Lake Superior provide a huge energy subsidy to the nearshore benthic zone that supports many organisms over winter, including lake whitefish, and provides an ecological link between habitats and food webs. So what is this talk all about? So today, Ralph and I are going to be talking about some ongoing work that we're leading to better understand Cregonian migrations in the Great Lakes and inform conservation and restoration efforts within the Cregonian restoration framework. Specifically, we're going to be discussing some projects that focus on cisco, Coregonus artedi in lakes Huron and Michigan. And I'm going to begin by talking a bit about a project that I'm leading to investigate river spawning by cisco in Great Lakes tributaries with a focus on the Spanish River in Ontario, Canada. As you can see here, this was a highly collaborative project. So before I get started, I'd like to thank all my co-authors on this work for their efforts. I'd also like to acknowledge that this work was funded by the EPA Great Lakes Restoration Initiative. So my colleagues and I started this project on river spawning because we felt that it was and still is an area where we have a lot to learn for Cregonians in the Great Lakes. We do know that Cregonians historically spawn in rivers throughout the basin. For example, cisco and whitefish used to spawn in the Maumee River up until the late 1800s, which is kind of wild for me to think about because of the river's current issues with things like eutrophication and algal blooms, as you can see in this photo here. We also know that connecting channels like the St. Mary's River currently support Cregonian spawning aggregations and are likely key habitats for Cregonian populations and communities. And there are also some recent good news stories about cragganines in spawning in rivers, like the resurgence of river spawning by Lake Whitefish and Green Bay tributaries that I mentioned earlier. But the extent of cragganine river spawning overall is not very well understood. And we haven't had any evidence of cisco spawning in Great Lakes tributaries in the last 140 years. So our objectives with this study were pretty straightforward. Essentially, we went searching for river spawning populations of cragganines. And in this case, we were sampling the Spanish River in Ontario, Canada. And if we found any spawning cragganines, we plan to provide baseline demographic information to describe them and compare their morphology and genetics to lake spawning populations. So you might be asking, why did we choose the Spanish River? For one thing, this river is located near the capture location of a potentially unique form of cisco called Manitoulin cisco, or C. artedi manisolinus. These fish were described by Walter Keltz in the 1920s as being relatively deep-bodied, somewhat short and darkly pigmented, especially in the head and sometimes in the fins. The Spanish River is also a large river without any natural or artificial barriers for 52 kilometers until it hits the dam in Espanola, Ontario, which means that there's a decent chance that it contains good spawning habitat for cragganines. Speaking of habitat, the river contains seemingly good spawning habitat up near that dam, including large rocks and cobble. And finally, there was one cisco caught up near that dam by Chris Davis and his crew in April of 2017. So we thought that it might be possible that cisco would also use this habitat in other times of year, like during the spawn. So just to orient you all, the Spanish River flows into Lake Huron, which you can see here. And specifically, it is a tributary to Lake Huron's North Channel, which is the region of these, north of these islands here in the northern portion of the lake that connects Lake Huron and Lake Superior. So if we zoom in on that region that was highlighted by that square in the last slide, we can see that the Spanish River flows into what's called the Whalesback Channel, which is a secluded little chunk of water within the North Channel. There's also an embayment within that Whalesback Channel called Aird Bay. And I'm bringing this up because I'm going to be mentioning both Aird Bay and the Whalesback Channel later in the talk. So just trying to give you a frame of reference here. So we conducted our sampling right around where this star is located, just downstream of the dam in Espanola, Ontario, which is, again, 52 river kilometers upriver from the mouth. And if we zoom in one more time, this time on our specific study site, on the lower right of this image is the town of Espanola. In the upper right is the dam. And we sampled the stretch of river that's between the two purple lines that I just threw up on the slide there. This is about a 1.5 kilometer stretch of river. We sampled via night electrofishing using this really old boat, which is actually a couple years older than me, and which I believe is the oldest Smith Root electrofishing boat that's still in service. We sampled from November 14th through the 16th in 2022, and we started well after dark, usually around 7 p.m., and continued for about 2 to 3 hours each night. And I'm going to jump right into some results for you all. As you can probably guess from the title of this project, the biggest and most important result in my mind is that we found cisco in spawning condition in the Spanish River. And you can see my co-presenter there, Dr. Tingley, holding one of those cisco. In fact, we found quite a few of them, and all of them were mature and had ripe gonads. More specifically, we caught 31 cisco over 3 nights, with 0 being caught the first night, 2 being caught the second night, and 29 in the third night. I'm really confident that if we had stayed and continued sampling, we would have caught a lot more fish. But since we're in the business of conservation and restoration, we didn't want to kill a whole bunch of them. So we just took as many as we felt we needed to get some baseline understanding of their biology and ecology. You'll note that over this time period, water temperatures were dropping rapidly. This was the first real cold snap in the late fall of 2022. And the fish started showing up as those temperatures dropped toward the typical spawning temperatures of around 6 degrees Celsius. Our 31 fish ranged in total length from just under 300 to just about 400 millimeters. And using otoliths, we estimated that ages range from 3 to 9 years, which is a relatively broad range. And importantly, there was a lot of age variation within the sample as well. So the most dominant age was age 6, which comprised half of the age sample. So this appears to have been a strong year class, but we also saw relatively high numbers of other ages, including ages 5 and 7, which would suggest that recruitment in this population may be relatively consistent. And finally, our sample was male dominated, which is common for cisco during the spawning period. As I mentioned a couple slides ago, all the individuals we sampled were mature and in spawning condition, and the eggs on the females were all ripe and loose, suggesting that they were indeed preparing to spawn. And I thought it would be good to just show you a few images of some of the fish we caught to give you a sense of the morphological variation in this population. So this is an example of a relatively large gravid female. As you can see, this fish was quite deep bodied for a cisco, in part because it was full of eggs, and it has some darker pigmentation in the head and on some of the fins. Here's an example of a relatively large male. And this fish, in my mind, is kind of the perfect example of what Kelps described as Coregonus artedi manitolinus. It's relatively deep-bodied for its length and for being a male. And it has very dark pigmentation in the head and the dorsal region and also in the fins. So this fish, when we pulled it out, was really striking to look at compared to most of the cisco that I've seen. It was like its fins were dipped in black ink. We did see a good bit of variation in this population. So here's an example of a much more slender male that is more average sized, about 325 millimeters. This one had some of that darker coloration, but not nearly as much as the last one that I showed. And here's an example of a slightly more slender and more average sized gravid female. Again, this one has some of that darker coloration, but this individual wasn't quite as deep bodied as that first female that I showed. And so to quantify this variation and compare morphologies of the Spanish River fish to fish from other populations, we conducted a morphological analysis using the measurements described in the Eschenroder et al. 2016 monograph on ciscos in the Great Lakes and Lake Nipigon. Specifically, we measured 9 linear morphometric characters, and we counted the number of gill rakers on the first arch. We then used a size correction approach, of which I'll spare you the details here, but I'm happy to answer questions if you have them at the end of the talk. So in general, we found that Spanish River individuals were similar in morphology to nearby populations. So what I'm showing you here is a principal component analysis of all of the measurements that I showed on the last slide. And the 3 groups here are the Spanish River fish in orange. In green, there's a recent collection of fish from Artedi Bay, which is that bay that I showed you earlier that's just outside of the river mouth. And these fish were also collected during the spawning period. And then there's some non-spawning period fish that were collected in other areas of the Whalesback Channel, again, just outside of the Spanish River mouth. So in general, what we're seeing here is a lot of overlap, morphological overlap, although I would like to highlight these 4 individuals on the far left extreme of PC1. So these came from the Spanish River, and they appear to have a bit different morphology from the other fish in this sample. And I'm going to talk about them a little bit more in the coming slides. So we wanted to put this variation in context, and so we asked the question, how do the morphologies of these fish compare to those of cisco from other parts of Lake Huron? And to address this, we compiled morphological data from additional cisco populations from around the lake, specifically from Government Bay in the Lachino Islands in northern Lake Huron, from the western end of the Bruce Peninsula, which we're calling Nawash cisco, and from Perry Sound in Georgian Bay. So I know this is kind of a busy figure, but I'm going to walk you guys through it. What I'm showing here are the same 3 groups you saw in the previous PCA, with the addition of Government Bay fish in pink, Nawash cisco in brown, and Perry Sound in gray. So you can see that in general, the Spanish River, Artedi Bay, and Whalesback Channel fish occupy this upper right portion of the figure and are pretty morphologically distinct from other populations. However, those 4 outliers from the Spanish River appear to be more morphologically similar to fish from other populations like Government Bay or Nawash. And just for reference, these two individuals that I showed you earlier that were more slender than some of those other individuals in the sample are 2 of those 4 outliers. And you can compare them sort of visually to this individual that I showed earlier, which was one of the ones that was in the far upper right portion of that PCA, was very much in that area that was distinct from the other populations. And I'll just quickly note here that these Spanish River fish mostly align with Coregonus artedi manitolinus, which fell in the upper right region of a very similar PCA conducted by Eschenroder et al. In 2021. So it does appear that these Spanish River fish are morphologically similar to that potentially distinct form of Manitoulin cisco. So to wrap things up here, we believe that our findings provide strong evidence that this was a spawning run of cisco in the Spanish River. First of all, the fish were mature and in spawning condition. The study site was 52 kilometers upstream of the river mouth, so we think it's unlikely that they would move that far upstream during that time of year, only to return to the lake to spawn. We saw that catch rates increased as water temperatures dropped, which is what we would expect during the beginning of a spawning run. These fish occupied habitats that we would expect them to use for spawning, like rocks and cobble. And finally, this spawning run had very similar phenology to other northern Lake Huron cisco populations. So taken together, we think that this is the best evidence of cisco spawning in a Great Lakes tributary since the 1880s, which is kind of wild to say, but it's very exciting. And another interesting wrinkle here is that this section of the Spanish River was formerly listed as an area of concern due to pollution from logging and mining activities, sewage treatment plants, and a pulp and paper mill in Espanola. So work has been ongoing to restore this ecosystem, and it's now listed as an area in recovery. So much like with the whitefish spawning runs in Green Bay that I mentioned earlier, the fact that these cisco are using this habitat for spawning provides evidence that Corregionines can recolonize previously degraded habitats. We also found that there was considerable age variation in the sample, and this may indicate that the population has more consistent recruitment success than some other cisco populations, such as in Lake Superior, where cisco are notorious for displaying boom and bust recruitment cycles. And finally, we found some interesting morphological variation in the sample. Most fish appeared to align with the Manitoulin form of cisco, but some fish were more like fish from other populations. So this could just be natural variation. Or it's possible that multiple groups of fish are using this same spawning habitat. And we're currently doing some genetics to figure this out. And I'll talk about that a little bit more in a few slides. I think these findings have some important implications for conservation and restoration. For one thing, going back to the 3 R's, this population may represent ecological and potentially genetic diversity that's worth conserving. And again, we're doing some work to better understand that diversity right now. It's also possible that this population contributes to the broader metapopulation in the area via portfolio effects. And this is something I'd like to investigate in the future. This work can also help to inform restoration efforts for other river spawners, including areas to target for sampling and perhaps where fish restoration efforts might be most likely to succeed. And as I mentioned earlier, this spawning run provides more evidence to suggest that Corregionines can spawn in previously degraded habitats, which suggests that habitat restoration might be a really useful tool for Corregionine restoration more broadly. But you might be wondering, this talk is about migrations. Is this actually a migration, or do they just live in the river? And that's a great question, because while I think it's very likely a migration, as of now, we don't know that for sure. So that is why we are currently doing some acoustic telemetry work on this population. Specifically, we sampled fish last November, about a week later in terms of calendar dates than when we sampled in 2022. And in this case, we found that all the cisco we caught were ripe and running, so we can confirm that they are indeed spawning at this location. We tagged 29 fish, including 5 females and 24 males, and we deployed receivers throughout the river from the dam in Espanola down to the river mouth. So our goal here is to understand the spawning run timing of this population, as well as their general habitat use and spawning site fidelity. I'll quickly note here that the tags that we used have a battery life of about 3 years. So we're hoping to learn a lot from these fish that we just tagged over the next few years. And also these tags operate on the same frequency that is detected by all of these receivers that are maintained through the Great Lakes Acoustic Telemetry Observation System. So if these fish migrate outside the river to the North Channel or to the main basin of Lake Huron, or even to other Great Lakes, there's a pretty good chance that we'll be able to detect at least some of those movements and get a good handle on the scale of their migrations. Alongside this telemetry work, as I mentioned earlier, we're also doing some genetics, specifically low coverage whole genome sequencing on these fish to better understand their genetic diversity and potential uniqueness. And we plan to describe the trophic ecology of this population using stable isotopes. And finally, as part of this project, we're going to be sampling additional tributaries to the North Channel this coming fall looking for more Corriganine spawning populations. And that's all I have for you all today. So thank you very much for listening. And I will pass it along to Ralph, who is going to talk about migrations to a different type of habitat, specifically to drowned river mouth lakes. So Ralph, take it away.
Great, thanks, Andrew. So now we're going to travel west to Lake Michigan and discuss how Cisco are apparently using drowned river mouths and whether these fish are migratory or residents within these systems. But first, I'd like to acknowledge my co-authors for this talk. This is truly a collaborative effort, and without these folks taking on different leads, it wouldn't have been possible. A brief background on Cisco in Lake Michigan. In the first part of the 20th century, Cisco were an important commercial harvested fish in Lake Michigan, but due likely to overharvest, habitat degradation, and invasive species, stocks collapsed by the 1960s, and they were largely lost from the lake. So while Cisco were not extirpated, they were extremely rare in the basin in that late 20th century period. However, Cisco started to expand again in northeastern Michigan around 2011 as ecological conditions changed. And this can easily be observed in multiple data sets, including commercial tribal harvest, shown in this figure, as well as standardized fisheries independent gillnet surveys near spawning locations where the expansions first began in Grand Traverse Bay. So as Cisco abundance has grown, we've also seen a huge increase in angler catch in the establishment of a recreational fishery. So this has largely been centered in northeastern Lake Michigan near that Grand Traverse Bay area. Of note is that the large proportion of Cisco that anglers are catching are actually reaching much larger sizes than were historically reported in the lake, with anglers catching fish commonly around 19 inches or 500 millimeters in length. So this would suggest that these fish are reaching larger sizes faster than historically in Lake Michigan or currently in other Great Lakes systems. Likely contributing to this higher growth is the contemporary Cisco in Lake Michigan are largely feeding on fish, as opposed to more planktivorous diets historically in the lake and in other Great Lakes systems. Now, you can learn more about this shift in diets from a recorded webinar by two of my co-authors, Jory Jonas and Ben Tershak, and that's available currently on the Great Lakes Fishery Commission YouTube site. So as this fishery has grown in the main basin, we've also observed increased catches in some connected waters in Lake Michigan, specifically these unique coastal habitats known as drowned river mouths. So drowned river mouths are transitional zones between the Great Lakes and its tributaries, and they were formed by glacial retreat when melting water would pool behind glacial till until eventually carving channels to the main basin, creating these lake-like habitats. They often provide more productivity or more productive environments than the adjacent main basin of Lake Michigan, and this can provide seasonally important habitats for Great Lake fishes. For example, it could provide overwintering habitats for Lake Michigan yellow perch, which migrate in the autumn and stay until spawn the following year. It also provides spawning and nursery habitats for Great Lake fishes, such as the important prey fish alewife. Drowned river mouths are particularly common on the eastern shores of Lake Michigan. This is the region where Cisco have been expanding. There are over 24 drowned river mouth habitats along the eastern shoreline, many of which, as we see in this map, have large lake-like habitats that are navigable by boats. So of special interest to today's talk on Cisco migration is the northernmost DRM shown here, Lake Charlevoix. So Lake Charlevoix is the largest DRM lake in Michigan, with an area of 69 kilometers squared and 37 meters deep, or about 120 feet. It's largely an oligotrophic system, and it's connected to the main lake after passing through the smaller Round Lake system and the last portion of the Pine River. So it's in this connecting channel that we have seen the emergence of a popular seasonal fishery for Cisco. So during a short period in May and June, thousands of Cisco are harvested off this pier, providing anglers with a great recreational opportunity. While some Cisco are captured in other connecting channels in Lake Michigan, catches are usually an order of magnitude or more lower than what's observed on the Charlevoix Pier. It's often assumed these fish are in the channel and the surrounding area to forage, potentially as they move between Lake Charlevoix and the main basin. In addition to this strong spring fishery, There's also been a winter fishery that has emerged on Lake Charlevoix. So ice fishing for cisco has become a popular attraction in the lake in the last decade and a half, with dozens of shanties on the ice on most days. So anglers are rewarded with large cisco to take home, and guided fishing trips are becoming more common. Despite its popularity, we still don't know a lot about this fishery. Specifically, if these fish are truly migrants from Lake Michigan, residents to the lake, or some mix of both. This has relevance to the management of the fishery by the Michigan DNR, as bag limits have been adjusted in recent years under the assumption that these fish are part of that larger population in Lake Michigan. So any additional information on movement patterns and determining whether there's any evidence of a resident population or even a mixed population between the main basin and Lake Charlevoix could have clear management implications. And Lake Charlevoix isn't the only DRM lake where cisco are being harvested through the ice. Reports from other systems along Lake Michigan's shoreline are popping up, although it's often as bycatch when folks are fishing for other species. So understanding winter ecology in other DRM lakes can also be useful in a broader understanding of seasonal habitats for the species. But fish concentrated in these habitats over winter could also offer us an opportunity to explore questions related to the cisco recovery that's been occurring in Lake Michigan. So there are two primary hypotheses to how cisco might have been expanding in Lake Michigan. So it's postulated in a recent review by Claremont et al. The first is that what we could refer to as the single stock hypothesis. And essentially what this means is there was a small remnant population, likely within Grand Traverse Bay, that has expanded outwards from that area, reseeding and repopulating within Lake Michigan waters along the eastern coastline and beyond. An alternative to this hypothesis is that there could have been several small remnant stocks in Lake Michigan that have emerged as conditions have improved in the lake, potentially from southern Lake Michigan, northern Lake Huron, or even the connecting waterways. So while existing catch data does suggest that the single stock hypothesis is more likely, looking at genetic structuring from samples in DRM lakes could provide us with an opportunity to explore this hypothesis more directly while these fish are in these relatively easy to catch aggregations in these habitats. So with these ideas in mind, my co-authors and I received a small pilot study grant from the Great Lakes Fishery Commission to answer several questions related to cisco use in DRMs. The first is, are seasonal movement patterns of Lake Charlevoix cisco indicative of migrations from Lake Michigan, resident populations, or some mix of both? The second, is there any evidence of genetic structuring that would indicate unique stocks, either within DRM lakes themselves or between northern and southern Lake Michigan? And finally, what are cisco eating in DRM lakes in winter and early spring, as we lack much data on that particular period of time? So we're able to take advantage of two existing acoustic arrays in northeastern Lake Michigan to address our first question on seasonal movements of Charlevoix cisco. These arrays are supported by different studies funded by the Great Lakes Fisheries Trust, the Great Lakes Fishery Commission, and the Great Lakes Acoustic Telemetry Observation System. And these are used to study Carignan movement, which is an effort led by my co-author, Daryl Hardorp, as well as lake trout use of protected areas in northern Lake Michigan. So in the winter of 2022, we also installed 3 additional acoustic receivers within Lake Charlevoix that you can see in the inset map above. This would allow us to track those fish after they're released in winter months. There's also 2 receivers in that Round Lake channel complex. And essentially this operates as an acoustic gateway. So we can see as fish move to and from the main basin into Lake Charlevoix. So around the same time that we put in these receivers, we spent several days on the ice and planting cisco with acoustic telemetry tags in the middle of Lake Charlevoix. We did this using our traveling tagging station, which is a setup inside of a heated ice shanty and has continuous flow of water over fish during the surgery. We also had an aerated recovery tank on the ice, and fish were collected via ice angling and implanted with these tags. So we were able to tag a total of 20 cisco during this process, and we collected 20 more for gut analysis, stable isotope analysis, as well as morphometric measurements similar to what Andrew described, and aging of these fish. Additionally, we ended up with 71 total fin clips for genetic analysis. And we were able to increase the sample size using fish that were donated us from anglers that were on the ice all around us. And I'll just quickly note that the success of this event was really due to the work of one of my co-authors, Ben Tershak, shown here on the left, tagging fish, who did most of the legwork to make it happen. And because we were surrounded by these interested anglers, it was a pretty rewarding experience where we got to show off some of our work and what we were doing and why. Even my co-presenter, Andrew Honsey, managed to get his hands on a cisco that day. So before we jump into our results, I'll note that 14 of these 20 tagged fish appeared to survive long enough for us to track them within the lake and outside of Lake Charlevoix. 2 likely died as a result of tagging, and 4 were likely collected by anglers. This would suggest a tagging rate of between 70 and 90%, which is really quite good, albeit this is a very small sample size. So we were happy with the results of using these angled fish. So here we have a representation of the acoustic array in the main basin on the left and an inset map of Lake Charlevoix on the right. So we were able to track these fish for over a year, from the winter of 2022 and the spring of 2023. And as I play this animation, you'll begin to see changes in the colors of the dots in the receivers of Lake Charlevoix, which is indicative of fish movement. All right, so first we can start to see these fish moving along in Lake Charlevoix as those dots change colors, staying in the lake until May and June. And at that point, every single one of them moves out into the main basin of Lake Michigan, moving throughout the northeastern portion and Grand Traverse Bay until about October and November, when all fish move into an area of known spawning aggregations near Elk Rapids and Grand Traverse Bay. At that point, they exit and move back into Lake Charlevoix in the winter months. So overall, it does appear that cisco are using Lake Charlevoix as overwintering habitat. When we look across the entire set of 14 fish that we were able to track, we can see some other interesting points as well. The first is that there appears to be uniformity in the movement among these fish within this system. So here we have a location occupancy representation with Lake Charlevoix in yellow, the main basin in turquoise, and Grand Traverse Bay in purple. And what we can clearly see is that these fish are actually spending about 6 to 7 months of their lives in Lake Charlevoix, yet they're spawning with that larger aggregation in Grand Traverse Bay based on this telemetry movement data. We also see there's this strong site fidelity, not just for the area within the spawning area of Grand Traverse Bay, but to the non-spawning area of Lake Charlevoix. So all of these fish are moving back into Lake Charlevoix the following season in 2023. While this was somewhat surprising to us, this result actually aligns with a recent study out of Lake Ontario, where cisco had strong fidelity for spawning areas, but also non-spawning areas throughout the year. Oops, sorry about that. So again, the primary message here is that cisco appear to be part of a larger spawning population in Grand Traverse Bay and are using Lake Charlevoix as overwintering habitat. And we really don't see any evidence of a resident population from these data. So during the winter of 2022, while we were tagging these fish in Charlevoix, we were also collecting fish from 2 other drowned river mouths where anglers have reported catching the species in recent years. And this was to assess or address that question of whether we see unique stocks throughout the basin. And this included Portage Lake, which is about 1/8 the size of Lake Charlevoix and farther down the coast, as well as Muskegon Lake, which is about 1/4 of the size and quite a bit farther south. These fish were collected by our partners at NOAA, either directly or from angler donations. We ended up with 25 fish from Portage Lake and 13 from Muskegon Lake. A special thanks to Jeff Elliott, pictured here, from NOAA, who led the genetic clipping and the collection of this whole fish throughout these systems. For our examination of genetic structuring, we included 2 other sources of fish from Lake Michigan. The first was fish collected from the Ludington Barrier Net Monitoring Program in the main basin. We included these fish from the earliest part of their sample season, which was April and May, as this appeared to be a representative time period where fish from Lake Charlevoix still hadn't moved into the main basin. We also included fish, 41 samples from Grand Traverse Bay spawning aggregations that were collected in the fall of 2022, as we were interested to see if they were the same genetic stock as the apparent migratory fish collected in Lake Charlevoix. So we used a genetic technique called rad-seq to examine genetic structuring, which is really an elegant way of subsampling across the entire genome in a semi-random way to allow them to look for genetic structuring within a population. We then used a multivariate approach, principal components analysis, which Andrew talked about earlier, to try to visualize whether cisco grouped by collection sites. If there were any genetic distinction between these sites where we collected fish, we would anticipate samples from the same site would group together in multidimensional space. We also used another PCA that included not only these Lake Michigan samples, but samples from a nearby stock of cisco in northern Lake Huron near Boot Island. Our hope here was to compare how genetic structuring in Lake Michigan compared to a stock that we might anticipate was distinct from fish found in the lake. And these were collected by the Fish and Wildlife Service in 2015. All right, so what did we see for genetics? In a nutshell, we didn't see any evidence of genetic structuring across the fish collected in DRM lakes, Grand Traverse Bay, or the main basin in early spring. You can see on our two PCA axes here that we have what appears to be a multicolored shotgun blast of data as opposed to grouping by region. Now, when we add in the Boot Island data, what we can see is those Boot Island fish that are shown here in blue on the right cluster together around the first PCA axes and are distinct from everything else in Lake Michigan. So this analysis not only suggests little variation in genetics within the Lake Michigan population and really a tight clustering of diversity, but we see that this similar population within a relatively small distance compared to distance down the coast on eastern Lake Michigan has quite a bit more heterozygosity in its population. So all in all, the genetic data set appears to support the idea of a single stock hypothesis of expanding cisco populations in Lake Michigan. The final question we were hoping to address today is to see exactly what cisco are eating in these drowned rivermouth lakes during the winter. So cisco diets are vary by season, and based on what we know from isotope data and observed diets, they really focus on three invasive species: round goby in the spring before the lake becomes stratified or as it becomes stratified, alewife during the stratified period in the summer months into the fall, and then spiny water fleas as primary diet items as we move towards December. Now there's other diet items that make up proportions of diet throughout the year, but these are the general highest proportion diet items. Now, what we don't know a lot about is that period from January to March and really into early April. So these DRM fish allow us to look at potentially migratory cisco feeding during that time of year. So we look here at the mean wet weights of diet items as a percent of diets. And it does appear that fish are primarily feeding on alewife if available and other fish species if not. So this actually aligns with anecdotal evidence from anglers over the past several years out of Lake Charlevoix and Portage Lake. So we observed only fish in the stomachs. They were the only diet items. We didn't see anything else. And in Lake Charlevoix and Portage Lake, nearly the entire diet was alewife. In Muskegon Lake, where we only had 6 stomachs with fish in them, alewife were just under 50% of the mean weight. While other fishes like rainbow smelt, shiner species, and brook silversides also made up a substantial portion of the diet. We also looked at how full the stomachs were across the 3 DRMs using an index of fullness, which is really just a ratio of stomach weight to total weight of the fish. And the main message here is that cisco are eating well in Lake Charlevoix compared to the 2 other systems, with stomach weight approaching 1% of the total fish weight in some fish in Lake Charlevoix. So another way to look at fish diet beyond what's in the gut is through stable isotope analysis. Stable isotope signatures are the ratio of different isotopes of an element of the same sample, can tell us what fish are eating and potentially in which habitats. For example, in the Great Lakes, we might see a sample with lighter carbon indicative of feeding in a more pelagic or offshore environment, and heavier carbon indicative of feeding in a benthic or nearshore environment. The type of tissue from the fish we analyze can also tell us about the diet of the fish or what habitats it might be using at different time scales. For instance, the liver can tell us about the diet over the period of weeks leading up to sampling, while muscle tissue can tell us about the diet of the fish over months. And comparing them can tell us about potential shifts in diet. So while we're still in the process of running samples on all of our diet items and cisco tissue from Portage Lake, we did see some interesting results in Lake Charlevoix and Muskegon Lake from these drowned rivermouth fish. So here we have the mean carbon signature on the y-axis and muscle and liver values in blue and green respectively in the figure for both Charlevoix and Muskegon. So what we see is that carbon is lighter in livers in both systems relative to the muscle. Since liver tissues tell us about more recent diets than muscle, this would suggest that fish coming through the ice are shifting their feeding behavior. And it could be representative of more pelagic feeding in drowned rivermouths, or potentially the same type of prey fish that has a different carbon signature within those rivermouth habitats. So even though this analysis is still underway, what's particularly interesting is that we may actually be observing the shift in diet in another body part of the fish that we're examining using stable isotopes, the eye lens. This ongoing work is being led by our co-author, Lily Fetzer, at the University of Wyoming, who has used eye lenses to track changes in stable isotope signatures over the entire life of the fish. So the eye lens can be peeled back like an onion, which with each layer indicative of a different period of the fish's life. In the figure on the right here, the core of the eye lens is represented by 0, with each subsequent layer of the eye being a different period in that fish's life, up until the most recent period, which is the last eye layer, right prior to capture. So for these two fish in the figure, one from Lake Charlevoix and one from Muskegon Lake, that were very similar in size and likely about 3 to 4 years old, we can see some pretty definitive patterns. First, there's a period where the carbon signatures are not changing dramatically, perhaps through the first 1 to 2 years of life. Then a period when the signature declines to values that are similar to what we saw in the livers of those fish in Lake Charlevoix and Muskegon Lake in the winter months. And then back to a heavier signature before potentially starting another downward trend. So these changes could in fact be representative of transitions in diet associated with movement in and out of DRM lakes. The first dip could be the 2021 winter season prior to sampling. And the second dip that we're starting to see in both of these fish could be a new drop in isotopic weight associated with movement into these habitats right before we collected them for our study. So because isotopic signatures change with somatic growth of the fish, it seems as though DRM lake occupancy could actually represent periods of high growth for cisco. And Dr. Fetzer's team is looking into measuring the diameter of these eye lenses and pairing these data with age data and growth data from fish otoliths to try to better understand these patterns. So a few take-homes on this work. Lake Charlevoix provides overwintering habitat for Lake Michigan cisco to some extent. What's particularly interesting is they seem to be occupying these habitats for up to 6 to 7 months at a time. So it's not just going in and out like we might associate with a spawning migration, just a substantial amount of time within these habitats. They also appear to be part of this Grand Traverse Bay spawning aggregation, and we don't really see any evidence of resident spawning populations within Lake Charlevoix, albeit our sample size was relatively small. We also don't see any evidence of unique remnant stocks along the eastern Lake Michigan shoreline. And this does support that idea of a single stock hypothesis that has emerged since the mid-20th century as the species has expanded again. And finally, overwintering cisco appear to consume fish, most notably alewife, while in these DRM lakes, mirroring what they do in the summer months. In terms of next steps, we're continuing on with the stable isotope analysis, trying to disentangle a shift in diet versus a shift in habitat use. Our growth rates high while in overwintering DRM lakes is kind of our primary question, and we're hoping to pursue that more with Dr. Fetzer. Additionally, in terms of future directions, this was a pilot study and not that many fish. So we think there's room for increased monitoring of cisco that are overwintering in these DRM lakes. And it could not only support some of the conclusions we've come to today, but also give us information on how occupancy patterns might vary in other DRM lakes that are similar or dissimilar to Lake Charlevoix. And potentially more importantly, if we're seeing these fish overwintering in these really southern DRM lakes almost immediately after the spawning season, are they actually aggregating and spawning in nearby areas in southern Lake Michigan? So this could have potential ramifications for management and conservation of the species. So wrapping both of these talks up, just thinking about some final thoughts with migrations of cisco. So cisco are migrating from the Great Lakes to connecting waters for different reasons and in previously undescribed ways. So this is potentially for both spawning, like we see these migrations into river habitats, as well as non-spawning reasons, potentially for overwinter foraging. So understanding their use and diversity of habitat use can inform how we think about conservation down the line. Also, some populations are utilizing previously degraded habitats, so specifically that Spanish River population. And this might allude to the fact that more rivers could be have populations reestablished potentially as conditions improve, and could point to the value of conservation initiatives that focus on restoration. And finally, understanding these migrations and how they influence metapopulations, communities, and ecosystems is important for conservation and management, and probably requires some additional study. And with that, I'll say thank you for listening and sticking around, and we can probably answer a few questions.
Fantastic. Thanks very much, Ralph and Andrew before. Really nice job of presenting a lot of great information. I knew some of that, but not all of that. And I just really, really cool stuff. So for questions, as we have done before, please pop your camera on, ask a question ideally. You can also submit questions in the chat that we'll try to monitor. And let's first sort of direct questions for Ralph, just because I know he's going to have to potentially head off soon, although we did finish in good time. And so normally I have to ask a question first, but Robin is all ready to go. So look at this. All right, here we go. Robin, go for it.
So my question is for Ralph, so I wanted to make sure I added him before he needed to leave. So I think that's I love seeing that you guys were able to really show that it was from the one population and it radiated out and went down the shore. Do you has there been any sampling or any evidence of recolonization of these rivers? So Muskegon Lake and Muskegon River are Great Lakes area of concern, and there's been a lot of habitat restoration in the lake as well as up the river. So I'm wondering if you see any evidence of spawning happening in those areas.
So to the best of my knowledge, there isn't clear evidence of spawning within river systems in Lake Michigan yet. It certainly seems possible that might move forward. Matt Herbert from TNC and Jason Smith have been working on monitoring river systems for quite some time, looking for spawning Corriganines, whitefish, and cisco. And they've had a few hits in certain areas, but nothing in Lake Michigan that would be a slam dunk like Andrew saw within his talk.
Okay. And we're kind of related to that, Ralph. I know you've looked for historical evidence of use of tributaries. How about use of drowned river mouths? Do you think they used to do it and maybe we just didn't document it in the past, or this is a new behavior?
Yeah, it's a good question. I'm not sure we have the information to really say that, or I don't. Folks from the DNR might have a better handle on it, as they've monitored some of those populations for years. I had heard some folks I talked to while we were out there collecting fish mentioned back even in the 70s, even in like July, trolling for cisco. These are kind of really anecdotal within Lake Charlevoix, so I don't know how much we can point to that as evidence, but it's certainly a large lake system that could, temperature-wise, you think could support the fish year-round. So in terms of other drowned river mouth lakes along the coast, those range in conditions throughout the summer months. So if anything, in most, it would probably be utilizing these habitats in the winter months and then moving out into the main basin during the summer, spring, and fall. Kind of what we see in Lake Charlevoix, just as conditions shift and temperatures warm.
Great, thanks. Michael Wagner.
Hi, Mike.
Hey, Ralph. How are you?
Good to see you.
Wonderful data. I'm just fascinated. And I was, particularly the Lake Charlevoix stuff, it sort of brings to mind that fission-fusion dynamics might be going on in the movement strategy of the animal. And so there might be groups in other lakes that are also returning to those spaces in the wintertime. but then, joining together in single spawning areas. And those types of fission fusion dynamics have been studied in fishes and birds and some other systems. It's really interesting. Any plans to tag animals in other inland lakes or drowned river mouth lakes to see if that's going on?
Yeah, I mean, I've been talking, my co-author, Daryl Hondor, for the most part, and then Jory Jonas and Ben Tershak. We've been talking a lot about that. Like, what's the next direction here? And it could be trying to expand the telemetry network in Lake Michigan to try to look at potentially movement in and out of other DRM lakes along that eastern coastline. So now that we have these results in hand and some of the genetic data back, I think it gives us a good leg up to think about that as a new direction.
Yeah, I might reach out to you because I'm working in White Lake and River and on some other stuff, and
Oh, cool.
I'm gonna chat with you.
Sounds good. Thanks.
Yep.
So kind of related to that, Ralph, do you have a sense that it gets too warm in like if those river mouths are so productive and so presumably more productive than open Lake Michigan,
Yeah.
is it just not enough depth, not enough thermal habitat there for them? But they're moving sooner than you think that it would be too warm for them, right?
Yeah, so it's really interesting. With these drowned river mouth lakes, there's a recent paper, I think last year was published, that showed they kind of go from oligotrophic in the north at Lake Charlevoix down to more mesotrophic in Muskegon Lake and further south. And within that paper and some other work, you can see that temperatures in some do warm outside the range that we would expect to be suitable during the summer months. However, thinking back to the perch work that I mentioned previously in the slides, that seemed to be some good documentation of unique stocks from Lake Michigan moving in simply to occupy for foraging until the spawn. So we could be seeing a really similar pattern across these drowned river mouth lakes with potentially very little establishment or continuation in residency through the summer months, regardless of those conditions.
Just going to build off that really quickly, Ralph. Yeah, I wonder if, like Bo said, that they seem to be moving out in, was it May, June, somewhere in there?
May to June, yeah.
Yeah, so, you might expect in a system, especially like Charlevoix, with that amount of depth and the oligotrophic state, that they might not have to move out temperature-wise until like September or July or August or something like that, somewhere in that ballpark. So I wonder if they're just following the alewife, like are the alewife moving out and they're moving out with them or something along those lines?
Yeah, so I think there's some questions around the alewife specifically. I was talking with Thomas Hook via email earlier this week, and just that, we still don't, in most of the DRMs, have a really good handle on the movement of alewife, or whether these are resident fish, or whether they're born near the drowned river mouth and spend the first year of life there and then move out. He did mention that in Muskegon Lake, they actually don't really have any records of overwintering alewife within that system. So the fact that we saw 50% of the diet items, albeit a very small number of stomachs, was really interesting and might maybe change how we think about some of these things.
Yeah, very interesting. And unfortunately, those alewife are going to be a little harder to tag than the cisco.
Yeah.
People are probably following the chat. Yuchen Cow said, based on some, I believe, commercial catch records into the 30s, showing Grand Traverse Bay, I think, was one of the higher areas of commercial catch. But then there were some surveys in the 1950s and showed no cisco there.
Yeah.
And then he said they surveyed Lake Charlevoix in the 1950s, but no cisco were caught. So it's interesting they're using those locations now. And Amanda asked about the timing of those surveys. It was spring to fall, but as you got into some of the seasons where you've been sampling them, seeing there, they did not have those. Do I have that right, more or less, Yuchen?
Yes, Rene knows.
Yeah, and wasn't Grand Traverse, I mean, I remember when we found the the relatively high catch rates in Grand Traverse based on your analysis of the commercial catch data.
That's in the earlier era.
Late 30s,
They had,
yeah.
yeah, they had 3 or 4 very good years, but they were also like the first place where the fisheries collapsed.
Yeah.
Yeah, so I do think just exploring this and thinking about how these habitats are being utilized down the coast could add a lot to our understanding of, it's kind of an emerging use and diversity of habitat. So we'll see.
Ralph, do you know, are there other drowned river mouth like lakes elsewhere throughout the Great Lakes that might sort of have
Yeah.
analogs?
There are, they're not as common as along the eastern shoreline of Lake Michigan. And I won't pretend to know where, but I remember when I was looking into this initially, they certainly do occur in other areas or similar types of habitats, but not necessarily in the numbers that we have.
I'm just thinking about like the large cisco population in Lake Superior, and if they are sort of moving into these, well, rivers or sort of associated embayments or,
Right.
I don't know. It's interesting.
So that's a good point. Like we're talking about drowned river mouths, which are pretty unique, and but it doesn't stop similar foraging movements to embayments or just river plumes throughout the winter months that could just have high productivity. So
yeah, one potential analog in thinking about that question that Bo just raised is not exactly the same because it's not a drowned river mouth, but you could look at South Bay in Lake Huron. Ralph, it's got one of the best data sets of, well, period, in the Great Lakes for a lot of things, very long-term data set, but there are spawning aggregations there of whitefish and cisco that might provide some interesting, comparisons, contrasts. Again, I believe I'm correct in saying that they are using those habitats for spawning in that case, but I'm not sure how long, we can look back at Recon, but I'm not sure how long, if they're moving out of those habitats in the summertime into the main lake versus staying in the South Bay itself, if that's just in the early life, and if as adults they're migrating in and out a lot more, or if they tend to stay in there longer. But that's a system that's pretty large and relatively oligotrophic and deep that could provide some interesting, I guess, comparisons to Charlevoix.
yeah, it's definitely worth looking into.
I mean, even Green Bay we can think of as like the largest drowned river mouth, right? In this place where it was very productive for cisco historically in Lake Michigan, but yet they stayed there and they spawned there, and we don't really know exactly how much they moved in and out, although it's possible they could have completed their entire life cycle there, at least up in the northern part, until things got really too productive. But is there spawning habitat in Lake Charlevoix? Like if you were to get some strayers, if that spawning fidelity were to become loosened and you were to get like somewhere just to say, hey, I don't know much about the habitat potential for them to stay.
So I think we have Ben and Joy. Oh, there's Joy. She just popped on camera, so I'm going to pass that over to her.
We've looked at this for lake trout before, and again, with cisco, and in particular these cisco, do we really know what their spawning habitat is? We have some guesses about places where we found them. It's not great lake trout habitat if you're going to look at an analogous reef structure. There's a few places where there's rocks. We've been underwater looking around a lot. It's a lot more sand and less rock than the main basin. There's a few places it could happen, not deep interstitial spaces though. I don't know if that helps.
No, it helps a lot.
Yeah.
No,
Yeah.
definitely. And the place where they're coming back to, do you have a like in Elk Rapids where you see them all them all coming back to there? Is it the reefy habitat, or is it more general than that?
Right, it is the reefy habitat, but again, you're into this thing where have we looked everywhere? They're not. Is this just where it started? We're in the middle of a population build and recolonization. Is that what a full, recovered stock is going to do? There's lots of spawning behavior questions underneath there. I think Tershak, who's also on this call, who shouldn't be, because he's on leave, but he has been doing some more detailed spawning observations for like whitefish and on reefs he's exploring. So he might have some insights to share here too. if he's able.
You're putting me on the spot here.
Yeah, I am.
I mean, we've seen cisco spawning on or seemingly ripe and running cisco spawning in places in Lake Michigan on the other side of the Leelanau Peninsula near Sleeping Bear Dunes on the same kind of habitat that lake trout and whitefish are spawning on. I don't know exactly what is making that habitat suitable. And from my experience in Lake Charlevoix, there's certainly lots of rocky shoreline habitat, but like Jory said, I don't know what the interstitial depth or what specifically cisco would be targeting. So it seems like these spawning populations are in other places, and I think that makes good sense considering how far south we're seeing them. And I don't think the fish that are in Muskegon Lake feeding in the wintertime necessarily spawned in Elk Rapids or in Grand Traverse Bay. So it makes sense that they're spreading around, but what habitats they're targeting for spawning, I don't.
Lots of interesting questions that end up in rabbit holes about recolonization and identification of new habitat, and what's going on here as it's building. So
maybe a question for Andrew Honsey. You sort of are suggesting that you think they're just coming into the rivers up into the Spanish to spawn and not being residents there. What conceptual model are you sort of operating under that you'd like to be able to test, or you think will be shown with the telemetry data you're going to have over the next couple of years?
Yeah, I mean, I don't know that I have a particular conceptual model other than that I don't think they were there. I mean, if they're in the river, they at least weren't where we started sampling that first night, because we didn't see anything, and I didn't think we were going to see anything at all. And then the second night we saw a couple, and then we saw a whole bunch. So they were clearly moving in. Were they just downstream before that? I'm not sure, but I think it's probably unlikely in my mind, just given the temperature habitat that would be there in the late summer. I would think it would be a little bit too warm for them to be in there year round, probably. It is a very productive river relative to the North Channel. There's a lot more food in there. There's a ton of fish in there. There's a lot of different types of fish in there. We were seeing a lot of bass and walleye, and there are muskie, there are sturgeon in there. There's a lot of stuff going on. So there's a lot more, I think, foraging opportunity there. And yeah, I guess it wouldn't surprise me if they overwintered there, or if they stayed there, kind of like what the Charlevoix fish do, except actually spawning there. Maybe they go out in the summer when it gets a little bit warm, and then they come back in. I'm not sure, but it's also possible that they just make the spawning run and they spawn and they leave. So yeah, I'm really curious to see what the telemetry data shows us, because I think that's very much an open question. But the river itself, I mean, there are plenty of holes in the river that are deeper than 40 feet. So I mean, it's not a shallow river, or at least not consistently shallow river. There are places where they could have refugia from some of those temperatures potentially, although it wouldn't stratify obviously in the same way that a lake would. It might still be cool enough in some of those deep holes for them to stick it out if they needed to. It's also interesting, I didn't mention this during the talk, but that one fish that Chris Davis's crew caught up near the dam in 2017 was 133 millimeters long. So I mean, it sounds to me like that's an age 1. It's not an age 0, and it might be an age 2, but it's probably not an age 3. So that's a young fish. Did that fish migrate, or did it just hang out there for a year or 2? And then when they mature, do they start migrating? So these are questions that I think are really interesting. Harder to answer because we can't tag the larvae in the same way that we can tag the adults. But yeah, I think there's a lot of open questions there that I'm really interested to try to figure out. And hopefully with the sampling this fall, maybe we'll find another population or 2, and we'll be able to compare and contrast, or maybe not. Maybe that's the only one up there. So yeah, we're going to find out.
So it's possible then just to extend that conceptual model into the earlier life history stages, where of course you have strong evidence they're laying eggs, larvae are hatching there. They may or may not be advected down into the channel. Maybe there's a way for them to sort of find more nursery habitat within the river proper. The fact that Chris found that fish in that same region as a yearling could suggest they could be there like other salmonids as juveniles, and then migrate out into the big open waters as adults, and then ultimately come back. Yeah.
Yep. Exactly. Yeah. And there's, I mean, we had a couple of tanks going, we had well, I guess a couple of recovery tanks, a pre-surgery and a post-surgery recovery tank going on when we were tagging this past fall. And we looked in those things about, halfway through as we're pumping water through them, and they were chock full of amphipods. I mean, they were everywhere in there. So I think there's a lot of foraging opportunity, especially for like, yearling or late age 0 cisco. When they're getting to those gapes where they can take advantage of those sorts of prey items, I think there'd be a lot there for them. And then it's just a matter of, whether or not they make those migrations out into the channel or out into the main basin, presumably for some other habitat that would be beneficial to them.
So a lot of food there for those young cisco if they were to stay. You saw the more consistent recruitment, but there's probably a lot of predators up in there too.
I would think for a small cisco, yeah. I mean, like I said, so that pulp and paper mill that's right there, which actually just closed down a couple months ago. There was an incident, I think it was about 30 years ago, where there was some black liquor released out of that pulp and paper mill. So that's the byproduct of the milling process, and it's very toxic, and it ended up killing basically everything down to the mouth, is what the locals told me. So that company has been doing some restoration efforts, including muskie stocking at that location. There's a bunch of muskie in there for sure. We've seen a few shocking them up. And you don't need a whole lot of muskie per unit area to have a lot of predation pressure, as anybody who's seen a muskie eat something understands. But yeah, one other, I mean, alongside that, so these fish are here, but people aren't actually fishing them during this period. So I think part of the reason people didn't know the cisco were there is because as part of this habitat restoration effort, there's a fishing sanctuary, a closed season from like October to May or something like that. So nobody's had lines in the water and wouldn't have caught any of these cisco, or wouldn't have caught any of the muskie that might be eating the cisco. So it's kind of a perfect storm of opportunity, I guess, for cisco to come back and utilize this habitat. And I will say too, really quickly, that there were anecdotal reports, nothing in the literature, but anecdotal reports of these fish being there many decades ago, like in the 50s, 60s, 70s, including in some really small tributaries to the Spanish River, like tributaries that you could jump across. They used to just stick a bucket in there. They didn't even need a net, and they could pull the cisco out. So yeah, lots of interesting things to be discussed there. But yes, muskie, walleye, bass, lots of stuff there. I see Ben's got a question. I know I've been talking for a while, so I want to give him a chance to ask his question.
Great talk, Ralph and Andrew. That was awesome. I was more interested in some of the telemetry stuff for the Lake Michigan's cisco. For some reason, I just pictured when the cisco left Lake Charlevoix, they'd kind of hang out closer to shore, and then then hang out in Grand Traverse Bay for most of the year. But it looked like quite a few of them went out to the Northern Refuge and were swimming around. Is that consistent with, I don't know if you know this, or maybe Daryl Hondorp will have a better idea if he's on the call, but are cisco from that kind of overwinter in Grand Traverse Bay are doing the same thing, or are some cisco in Grand Traverse Bay just living there the whole time?
Yeah, if Daryl is on the call, I don't know. Daryl, do you want to pipe in and provide some insight, or I can try, I guess. Might have lost him. All right, I'll start talking. Maybe you can pick on. So I think that work's ongoing, Ben. Trying to figure that out. And Daryl's been doing a lot of work on tagging cisco in that region. And when he saw these data, he wasn't surprised about the movement out into the northeastern area of the lake. But I won't speak to the movement patterns he's observed yet. I do know that it was pretty diverse. In fact, he can't unmute, so maybe I'll have another answer in the chat. Or maybe there's a way to do that from your end, Renee. I don't know. Okay. Anyway, so I think Daryl can probably provide some more information in the chat on some of the results he's been seeing, but I know it's kind of the diversity and movement of those Grand Traverse Bay fish, from my limited understanding.
Or even, is there any, I don't know how many like summer agency surveys occur in the refuge area, but do commercial fishermen, catch cisco outside of the bay in the summertime or anything like that? Is it, if anyone knows? I don't know.
Jory's got her hand up. She might actually have some insight on this too.
Probably not along that train of thought. I don't have evidence of individuals being collected by anybody out there. You know, I think Jason Smith would probably be, or somebody from Little Traverse would be better to weigh in on what the commercial fisheries are picking up. I know they do pick them up though. What did they had 3,000 pounds of fish. So I guess I can say that, yes, they are picking them up out there. The other observation I wanted to make is that Ben Tershak's leading the paper. I'm a co-author with him, and we are about to submit, and I think you heard about it in our talk months ago. Cisco are basically Chinook salmon. And so I've started to think of them that way. And they're going to be up pelagic, roaming around and eating alewife. And so when you think of them that way, this becomes less surprising.
Yeah.
From where I'm sitting. So we have some new information from the isotopes that are helping to reinforce that where there's fallout in the food chain of Lake Michigan. And Ben, I don't know if you want to add anything to that, but I just think it's an interesting sideline of evidence in support of what we're observing, you know?
Yeah, you're putting me on the spot again. Yeah, I think in Lake Michigan, we for sure see Cisco, you know, moving around a bunch on the telemetry side of things. If Daryl could unmute, I guess he's got some stuff in the chat here, but definitely from a stable isotope standpoint, you see like really similar isotopic signatures that look really similar to other pelagic sort of open water predators, which seem to suggest that they're probably moving around, taking advantage of prey caught, or that they're you know, taking advantage of similar prey sources as those pelagic predators, and suggesting that there's a high degree of mixing and movement. But I think I'm just looking at the chat here with Daryl. Yeah, Daryl has a project that's ongoing looking at this exact question. So yeah, I don't know. Does that sort of answer or like address what you're talking about, Tori? I mean, I think I'm just echoing what you said.
Yeah, exactly. Echoing, or if you had anything to add, you know, now that I'm engaging my brain here, we definitely have commercial records through time from out there. Again, it's mostly through the tribal fisheries. And then this observation about their feeding behavior and basically being Chinook salmon, it becomes less surprising if you think of it more like that and less like a lake whitefish.
Good question, Ben. And I think there's also some telemetry data in northern Lake Huron. I don't know if it shows the extent of sort of that offshore distance from shore as some of these other studies. And I don't think we have the isotopic data from Lake Huron to sort of say that they are as piscivorous as this Lake Michigan stock. But you also saw in Ralph's data there from Amanda the quite different genetic structure between at least the Lake Chinook, Lake Huron, Cisco, and then everything in Lake Michigan. So.
Hey, Bo, it's Daryl. We finally got this straightened out. It turns out that I had to agree to being recorded in order to have permission to unmute. But yeah, pretty much everything's been said. We have a ongoing project looking at Ben's question. So I don't want to take up any more time, but people can get a hold of me if they're interested in more about that.
Thanks, Daryl.
And Bo, I can comment quickly on at least what I know about the Lake Huron adult Cisco stuff from the Lake Chinooks. I don't want to speak too much on this because this is, you know, Todd Hayden's project, and he would know a lot more than I do. But my recollection is that they spawn in the Lake Chinooks Islands. These were, I think, primarily government bay fish spawning in the Lake Chinooks Islands, and then they, I believe, overwintered there and then kind of moved out in the summertime, more like June, July, than say, I think, May. So it was a little bit later, and I think the idea was that it was probably more temperature driven. But then they come right back, and so they spend a fair amount of time near shore, and then they move off. And you know, he had a pretty extensive grid. They were moving out into the main basin of the lake, you know, pretty far, but obviously it's a huge lake, and we at that time the course grid that's now covering the entirety of the lake was not in place. So we can't tell, for example, if they were moving to the south end of the lake and then back or not. But they were well out into the main basin for at least a few months.
Yeah, thanks for that. Elvita has a comment there talking about, I don't know if you want to unmute Elvita and talk about your study with regard to subsidies.
I just I wanted to write, but no, there was that we were discussing alewife movement, and we recently published the stabilized dope study also with Ben and Thomas Hook, where we were measuring isotope signatures in alewives in Lake Michigan and 3 tributaries like Muskegon Lake, Trail Creek, and San Joseph. And we really found out that alewives were strongly subsidized in nearshore Lake Michigan by those tributary resources, and vice versa in tributaries. We also found a strong lake signal that suggests that alewives are moving, and yeah, so that fish is using this tributary habitats. And because before there was this discussion if cisco is following to alewives and to feed on alewives, so yeah.
No, that's helpful.
Yeah.
These fish are finding these unique and productive habitats.
Yeah. Thank you.
Thank you. Well, I'm looking at the time. Any other burning questions out there? This has been a great discussion, and we really appreciate the time that Ralph and Andrew took to compile a great presentation for us. Remember again to register for next month's webinar, looking at some laboratory work of potential bottlenecks for whitefishes in Lake Constance. And we do have a few more we do have a few more openings. If any of you would like to volunteer yourself to join one of these to give some of your research. A few of these months left, and then of course we're wide open into 2025 as well. If you think you're going to have something a little bit farther out, but you'd like to get on our calendar, we'd welcome that too. Reach out to Renee or myself if you're at all interested, or just to learn more about that opportunity. Seeing no final questions, you can give a virtual hand clap and or send a quick chat to our authors if you are so inclined. And thanks again for joining us. We had a great turnout, and thanks for the great discussion and questions to everybody. We'll see you next month.
Thanks everybody.
Thank you, everyone.